Hello everyone, we're the EpiNOD team, and in this video we're going to explore the wonders of chromatin dynamics and epigenetic reprogramming during the nodulation process in the leguminous model plant Medicago Truncatula. Our project context involves addressing the environmental impact of agriculture and eventually contributing to more eco-friendly farming practices that reduce the negative impacts of synthetic fertilizers. We expect to provide fundamental knowledge about the molecular mechanisms of the nitrogen-fixing symbiosis that Medicago establishes with a bacteria called Rhizobia. This bacteria provides nitrogen to Medicago in exchange for carbon. This exchange takes place within specialized organs known as nodules, which form on the plant's roots. During nodulation, chromatin dynamics can be influenced by various factors, including genetic and epigenetic mechanisms. Epigenetic modifications involve changes in gene expression without altering the DNA sequence. For example, histone variants can affect DNA coiling, leading to alterations in gene activity. One group of genes influenced by this are NCRs, which are required in the process of nodulation. Another factor to consider are transposable elements. Transposable elements are DNA fragments with the ability to relocate within the genome. As they jump around, they induce alterations that may lead to diverse effects on gene expression. With this in mind, we mainly want to answer two questions. One, how does the arrangement of chromatin differ in host cells that are going through nodulation? And what are the observable epigenetic changes? And two, what is the impact of transposable elements on the expression of NCR genes during the nodulation process? Using bioinformatics, we've chosen NCR genes expressed only in nodules and are studying how histone variant modifications affect their expression. We're also looking at specific histone variants linked to nodulation, some associated with active gene transcription or euchromatin, and others with tightly packed genes or heterochromatin. Following this analysis, to be able to observe the dynamics of the NCR genes and histone variants, we will fuse them to fluorescent tags. These tags absorb and emit different colors of light, making them visible with confocal microscopy. When the gene is being active and transcribed, the fluorescent protein will also be expressed. Then, during the process, which is called hairy root transformation, we will transfer our design construct into a bacteria, which is able to insert its plasmid, including our sequence, into the plant's genome. After that, we will perform a chromatin immunoprecipitation, or CHIP, which uses antibodies that will specifically bind to our fluorescent tags, allowing us to understand the level of histone variant enrichment with the chosen NCR genes. We will also do a mobilome-seq, which is a technique that analyzes active transposable elements in the genome by isolating and fragmenting them from DNA, allowing us to capture live mobile elements. Then, using bioinformatics, we can identify, characterize, and understand how they interact with our genes. Overall, we aim for this project to contribute a valuable piece to the puzzle of symbiotic relationships. This insight could potentially lead to more efficient nitrogen fixation in legumes, as well as the introduction of the symbiosis into non-leguminous crops. Thank you for your attention.